Jai Hind everyone. I am Pankaj Kumar, and today we are going to read about the roles of antibody and the most interesting topic and the most important topic from which the question asks at least one questions, that is the hybridoma technology. Okay, so along with that, we will see the T cell. We will start the T cell maturation, T cell activation, and then in 1 hour how much we can cover you'll see in the next 1 hour. Okay, so before going to the class, I want to give you one information in the description box. Something is waiting for you, that is that free test series link. You can tap the link and you can try to solve the questions regarding to the immunology. Okay, so you can go there and tap the link and you can join the live classes by attending the by downloading the U Biology app. So what it is, role of the antibodies, it's a ninth lecture of the immunology, and we reached till now, we read so many things till now. And the role of antibody, to understand the role of antibodies we have to understand what is the humoral immunity first. So humoral immunity is the like fluid immunity, the immunity, the defense system which is provided in a fluid form. Okay, that is called the humoral immunity. Okay, so humoral immunity and the role of the antibodies. What is role of antibodies? We'll see now. Humoral itself means fluid. In fluid, so many proteins will be there which will help in the agglutination, pathogen clearance, opsonization. Like complement system is there in our humoral immunity, which will help in the opsonization and stimulate the complement system because to increase our immunity and the neutralization of the toxins. These are the important points about the humoral immunity. What do you mean by the role of the antibodies? Like antibodies are there in our body, and they are little diverse and specific according to the pathogen. So the immunoglobulins E, which will help in the allergy reactions. Like allergic reaction, whenever you will see the rise in the immunoglobulin E, it means you can understand there will be something wrong with your uh, allergy. Like that is a response of the allergy, like something happened wrong with your environment, or maybe internal it will be. Okay, so hypersensitivity will be results in the high increase of the IgE. What it will do, it will degranulate the mast cells, basophil cells, and then it will help in the release of the histamine. Once the histamine will be released, the more inflammation will be occur. Your blood vessels will become dilated, and the injury site will be become red, inflamed, swollen, little more heated, and more like WBC can reach there for the clearance of the pathogens. So it will provide the allergic reactions. Okay, IgE. So IgG is the most abundant, antibody. It is around 80% in our uh, fluid, body fluid, not exactly body fluid. We can say in all over the antibodies, all the antibodies, its percentage is 80%. Okay, it will help in the opsonization. Opsonization means what? I already taught in the last videos, but now again I am telling you what is opsonization. Opsonization is just a way of weakening of the pathogen. Means like uh, like a ketchup. If you're eating something like any nuts or any pakoda, then if the ketchup will be there, the taste will be increased. So if IgG will be there, so it will increase the taste of the what? Antigen. Antigen means what? Like pathogen cell. So if the taste will be increased, for whom? The taste is increasing for the macrophage, neutrophil. So the neutrophil and macrophage, like with the taste, they will eat this pathogens. Okay. So, so this is all about the IgG. Another thing, it will help in the activation of the classical complement. Its component, it's written complement system. So classical complement system. We will read it uh, in the coming lectures. Complement pathway. Okay, and the toxin neutralization, antibody mediated cytotoxicity by the natural killer cell. This is all provided by the immunoglobulin G. Immunoglobulin M will what it will do? It will activate the classical complement. Again, it will activate the classical complement system as like IgG, but like IgG, it will not help in the opsonization. It will not mediate the antibody cytotoxicity, and IgA will help in the neutralization of the pathogen, and IgD is the very least in the antibodies. Its percentage is around 0.5%, and it will help in the complement systems. Okay, this is all about the roles of the antibodies. Hope so you understood. So, hybridoma technology is what? Hybrid means what? The mixture of the two or more characters. Like a hybrid means hybrid animal. Hybrid animal means you mix the two characters of two different species character in a one animal. Okay, you mixed it. Hybrid laddu means what? You mixed like motichur laddu with the besan laddu. Like you will make another laddu. That is a hybrid thing. Hybridization you already studied. The mixing in the chemistry, the mixing of s orbital, p orbital. So that is mixing something. So here also we are doing the mixing, hybridoma. Hybrid we are making mixing. We are doing mixing of what we are mixing? We are mixing beta the myeloma cells. What is the myeloma cell? Myeloma cells are the cancer B cells. And what is the cancer B cell? The B cells which is like uncontrolled, which are growing uncontrollable, and it is making a tumor. So that is called myeloma. Why we want a cancer cell? That is the question. So the cancer cell have the unlimited growth. First thing, and if it have the unlimited growth, if we will induce a B cell, if we will make one type of B cell which will not exactly like a myeloma, but it will gain the uncontrolled growth, it will gain uncontrolled growth, then we can make so many antibodies and we can do the reactions, we can check the experiments in our lab. Otherwise, B cell have its own life and it cannot survive simply in the like as in the dish, like it will not survive in the experimental dishes and all. Okay, got it. So we want a cancer type of cell which can easily survive outside the cells also, and it will be uncontrolled growth. That's why we are making, we want B cells. Now, this one kind you seen, the cancer B cell you will take. Another cell, which cell you will take? Another cell you will take the normal B cells. Okay. Okay. So normal B cells, specific B cell you will take, which will produce a specific type of tissue. Before moving to the next, moving forward, I want to tell you about the monoclonal and polyclonal antibodies. You just read this line, pause the video and read this line what it is telling. Okay. Hybridoma technology used for production of more number or unlimited number of the antibody. This is our vision. Got it? Now, what is antibody? Before going to the hybridoma, we are seeing little a type antibody. Okay. Monoclonal or polyclonal antibody. So monoclonal antibody means what? The antibody which are made from a one type of clone. It is very important. Again, I am saying the antibody which is made from the one type of clone, that is called monoclonal antibody. Got it? So the monoclonal antibody means what? Suppose this B cell is there, and this I am giving this B cell as a X name. Okay, X its name. So this X B cell is specific for this site of antigen. Okay, I will make you different type of. Uh, I will make the different diagram only. Okay, so So you can see this X cell, X B cell is it is very specific for what? It is very specific for this type of antigen. It have the paratope. You know what is paratope and epitope. So antigen have the epitope and antibody will have the paratope. So paratope of the antibody will bind with the epitope of the antigen. So if you want to understand, you just please go through the all the videos which we uploaded already. Okay. So the paratope of antigen will bind with the antibody, and now this X cell will become mature. Now it suppose it become it was naive B cell, and now it become mature, and it become the plasma B cell. Okay, plasma B cell. Now this plasma B cell is little mature. Now we will make the mosaic of this plasma B cell. Suppose so it is like that. Whatever the plasma B cell is, this mature B cell, it have little this mosaic. Okay. So this why I want to say making this mistake, because the most of the time the profession of the parents will be followed by the profession by of their children. Okay, the professions are followed by the children most of the time, not every time. Okay, so here the B cell, this B cell is matured from what? This above X cell. Now, whatever kind of antibody it is making, the same antibody will be made by this, and that antibody will be again very specific for this. Antibody is very specific for what? This antibody is very specific for beta this type of pathogen, this type of epitope. Okay, this is very specific for this type of epitope. Got it? So it is forming same only one type of antibody which is very specific, that is called monoclonal, producing different paratope type of antibody. This I maybe wrote this is for the different thing. I am erasing this. Okay, I should erase it before. Now you understood the what is monoclonal. The antibody have the specific antibody have, wait just a minute. Okay, so whatever the clone you will get from this parent cells, now whatever the like descendant of this B cell will be present, that will form only this type of antibody. Okay, hope so you understood this type of antibody they will form. Got it? Now the thing is that polyclonal. Now the polyclonal means what? If you'll see, this is the B cell. It have the antigen. It can form the antibody against this also. Now, if it will recognize the or the other B cells which will recognize this B cell is recognizing an different type of antigen. Like if it is recognizing this type of antigen, now the antibody will be there. It will bind with this. Okay. So this is what beta. This have the different type of antibody for the different epitope. For what? For different epitope. So it is the clones of this B cells. The clone of this B cell will produce different type of antibody, different antibodies which are specific for the different epitopes. Okay, producing antigen antibody producing clonal cells clonal cells which clonal cell which produce clonal cell which will produce antibody which will produce antibody which will produce antibody for different different epitope of antigen different epitope of antigen. So this is what the polyclonal antibodies. Okay. Hope so you understood this anti monoclonal polyclonal. And now I will tell you why I taught this. Because we are going to take monoclonal cells. Monoclonal, we will make the monoclonal antibodies. For that we will take the B cells, monoclonal B cells. We will take this B cells. Okay. And this B cells we will take and we will introduce. Where we will introduce beta? We will introduce in the myeloma cells. Okay, now, so this is the mixing. I hope so you understood this concept. The mixing of the cancer B cells and the monoclonal B cell, we will call this as a hybridoma technology. You can ask whether polyclonal B cell can be introduced. Yes, it can be introduced. So how the experiment was first time it was done by the Milstein and the Coiler, we will see that experiment. Okay. So, are you ready? Okay. So the Milstein and GJF. These are the two scientists, those who first time those who made this hybridoma technology. We will see. We will see the way how they make the hybridoma technology. So this there will be one mouse. Suppose Okay, this is a mouse. First, what we will do, we will immunize it. Okay, what we will do, we will immunize it. Immunization means what? We will give them some immunogens or like attenuated anything like attenuated virus or something we will give, by which it will produce immunity against that. Okay, listen it properly. Very important it is. Okay, very very important. Hope so you are there and you are watching properly. Okay. So this is the way immunization we are doing. Okay. So first step is what? First step is immunization. Immunization means what? We will develop the immunity in it. Okay, we will develop the immunity in this mouse. How we will develop the immunity? We will uh, give some immunogens, or we can say we can give the some attenuated viruses and all. Now we immunized it. So what will be happen once a foreign molecule, like this is a foreign molecule, will go inside this? So this will be targeted by antigen presenting cell, and this antigen presenting cell will be what it will do? It will go to the T cells, and then T cells will go to the B cells, and this B cells will be mature there, or this antigen will be taken by the B cells, and finally by the T cell dependent B activation, it will be activated. Got it? There will be two way now. Either it can directly go to the B cell, and the B cell will go to the helper T cell and it will activate it. Or either it can go to the antigen presenting cell. Antigen presenting cell will be bind with the T cells, and then T cell will give the information to the B cells. I told you two methods, and I will go with the simplest method by which you will be not confused. Okay. So, so antigen came, antigen presenting cell will recognize, it will tell the B cells, T cell will activate the B cells, and now B cell will become the naive B cell will become the mature B cell. What it will become? It will become the mature B cell. Now it is matured. This whole process, this maturation and activation is occur in where? This is occur in the spleen. You know where it is occur? B cell activation occur in the spleen. We already studied. Okay. So this how it is occurring. You can go and watch the immunology lectures, which is already uploaded in the classes. Now, this spleen we will take. Okay, why we are taking this spleen from the mouse? We will dissect the mouse and we will take out this spleen, because this spleen have the B cell. Our important B cell it have. Okay, so that's why we took the this spleen B cell. So now this spleen is taken from this mouse, and we will what we will take? We will take the B cells. We will take the B cells. From there, we take the B cells. Which kind of B cells it are? These are the monoclonal type. Okay. Now in the other dish, in the one plate, okay, in one plate, I am saying dish. In one plate, we are growing what? We already having the we are already having some tumor cells. Which tumor cells it are? These are called as a myeloma cells. Okay. So these are myeloma cells. See, if you're thinking I'm going slow, you can fast forward your videos, and you can have a good experience of the videos. Okay, so and going slow is like okay for everyone. Now we will mix these both cells. Okay, B cells and myeloma cells. Okay, B cells and myeloma cells, and now it will be called as a, suppose it will be called as a, it will called as a hybridoma cell. What it is called, beta? Suppose it is a green color means what? It is hybridoma cells. So some of them are completely cancerous. Okay, are completely cancerous cells. Okay. Now this hybridoma, along with this, when we will mix when we will mix myeloma and the B cells, we will grow them the HAT medium. Okay, we will add some HAT. What we are going to add? We are going to add HAT. And what it is called? Hypoxanthine thymidine. Hypoxanthine thymidine. Okay, if you know the answer where it will be used in the which pathway it will be used, you can comment in the comment box. And if you don't know, we will tell you soon. So, hope so till now you understood and you may commented in your comment box that why hypoxanthine is required. So, ah, continue. So these are the hybridoma cell, and some cells will be there which is not transformed in the hybridoma, and that will be the myeloma cell. Suppose this is my myeloma cell, okay, and some spleen cells, this B cells become the hybridoma cell. The green cells are the hybridoma cells. I will write it here. Green cells are the hybridoma cell. Hybridoma cells will be there, and this already I told this is a myeloma cell. Red color cells are the myeloma cells. So why this is hypoxanthine we are giving beta? Hypoxanthine we are giving because this will help in the de novo synthesis. De novo synthesis. Okay. And what is happening in this de novo synthesis? The nucleic acid will be formed. Okay. Nucleic acid. Nucleotides. Usually the nucleotides will be formed. Okay. So this nucleic acid will be formed. If the nucleic acid will be formed, it will form the genetic material, the DNA. It will form what? It will form. It can form the DNA. No. Okay. But if the we have to segregate the hybridoma, so what we will do? We will stop this hypoxanthine. After this, we will bring this culture out, and we will provide in the same culture, that is in the same culture we will provide the aminopatrin. What will provide? Aminopatrin. And this aminopatrin is, first we will make some cells. Okay, this aminopatrin is what? My handwriting is not good, huh? Wait, let me write. That is true. Aminopatrin. So aminopatrin is what, beta? Aminopatrin is the analogous of the folate. Okay, it is analogous of logos of folate. So why we are very focused about the aminopatrin? Because we have to, because because we have to segregate the hybridoma and the myeloma cell. We don't want the cancerous cell, like we don't want the B cells. So if we don't want the B cells, we don't want the B cell. Sorry, we don't want the myeloma cell because it is a cancerous cell. We want only hybridoma cells. So we have to segregate this red and green cells. Red and green cells mean hybridoma cell and the myeloma cell. So how we will segregate? We will provide the aminopatrin, and now you just listen properly whatever I am writing here. The important thing is that in the presence of dihydrofolate reductase enzyme, the dihydrofolate usually the dihydro di DHF, okay, dihydrofolate I am writing it, will convert into tetra tetrahydrofolate in presence of what, beta? Dihydrofolate reductase. Dihydrofolate reductase. In presence of dihydrofolate reductase, reductase, it will form tetrahydrofolate. This is the normal condition of any cell. This is a normal condition of any cell, and then this tetrahydrofolate is required for what? It will form the nucleic acid. Finally, it will form the nucleic. It will form the nucleic acid. It will form nucleic acid. Okay. Now this nucleic acid will give the it can give the DNA file. This up to here everything is clear. Now until the DNA will be formed, the myeloma cell will grow and the hybridoma cell will be grow. So because DNA is very important now, up to here it is clear. Now, what I said, this aminopatrin, we have to give one shape to this aminopatrin. Suppose this is the shape of the aminopatrin. Okay, this is the shape of aminopatrin. This is our aminopatrin. Now this aminopatrin and the dihydrofolate is like this. Dihydrofolate is also looks like this, triangle only. Now this aminopatrin and dihydrofolate, if both are there, then the aminopatrin will go and bind this dihydrofolate reductase enzyme. So once this dihydrofolate reductase enzyme will be blocked or bind with this aminopatrin, it will bind with the aminopatrin, then then it cannot be formed. Understood? Again, listen properly. Before it, before it, the dihydrofolate is giving what? Dihydrofolate giving the tetrahydrofolate. Okay. But now what happened? The important thing is that the amino patrin is analogous to folate, and it will goes and bind to this DHF reductase. Once it will be blocked, then the tetrahydrofolate cannot be formed because it is because the aminopatrin is bind to the DHF reductase. And the same, these both are the substrate for the reductase enzyme, and this type of inhibition is called as a competitive inhibitor. What it is? Competitive inhibitor. Because this both can bind, but competitively, this aminopatrin is binded to the DHF reductase. Okay. Now, the question is arising like this. In the competitive inhibitor, the Vmax will increase or remain since, and the Kmax will increase or remain since. Please do comment on your box. Now, up to here, it is clear. Now finally, the THF will be not formed, the DNA will be not formed, and the problem will be created for the both cell. The hybridoma cells will be also not forming the DNA. Then the myeloma cell also not forming the DNA. But there is a nice choice for the hybridoma cell. Okay. So finally the de novo pathway here it is closed. De novo pathway will stop. Okay. De novo pathway will stop. So the hybridoma cell switch here you just write. So hybridoma hybridoma cell will switch the SIWT to salvage pathway. Okay, this is the two pathways which are there for the which are there for the what formation of the nucleic acid. Okay, so if the de novo pathway will stop, so the hybridoma cell will switch to the salvage pathway, but Myeloma cell cannot switch. Myeloma cell, but myeloma cell cannot switch. Okay, this is the important thing. Now, what will be happen? The hybridoma cell, this hybridoma, this hybridoma cell will be go for the what it will go. Now this part is finished. The myeloma cell cannot grow. Now we have to save this hybridoma cell by the salvage pathway. So the hybridoma cells have the HGPRT. What it have? It have HGPRT. This is the most important enzyme. This is the most important enzyme, which name is hypoxanthine guanine, xanthine, hypoxanthine guanine phosphoryl transferase. Okay, phosphoryl transferase. Now once the this enzyme will be there and hypoxanthine is already you are providing here, you already provided the hypoxanthine. If this enzyme will be there, hypoxanthine will be there, then it will switch to go for the salvage pathway, and only hybridoma hybridoma cell will survive. Okay, only hybridoma cell will survive. Okay, so this is survive in aminopatrin aminopatrin. Okay, this is the very important thing. And finally you will get the hybridoma cell, and now you can make so many antibody from this. You can make what better, you can make so many monoclonal antibody. Okay, produce monoclonal antibody. So up to here everything is clear. Now if you have any doubt you can ask me. I will tell you the answer for that.